Alright, so we're going to start talking about the process of photosynthesis. Alright, so first I'm going to lay down some groundwork connecting the processes of photosynthesis and cellular respiration. We'll talk about some basic terminology you need to become comfortable with, and we'll move on into talking about the overall big picture of photosynthesis. And I'll move on and talk about each individual process that's involved, and then some exceptions that you see in other plants.
The Earth is continuously bombarded with energy from the sun and radiates most of that energy as heat back into space, making Earth's biosphere an open system in terms of energy. In contrast, the Earth's biosphere is primarily a closed system in terms of water, minerals, and nutrients. There are finite amounts of these substances which are recycled through the living and geological components of the biosphere, called a biogeochemical cycle. Carbon, a key element of organic life on Earth, cycles through the biosphere in this way. A tree, like all plants, algae, and cyanobacteria, is an autotroph, an organism that produces its own food from inorganic molecules to facilitate growth. Through photosynthesis, photosynthetic autotrophs use energy from the sun to combine carbon dioxide from the atmosphere and water from the soil to form carbohydrates such as glucose. Oxygen is released as a byproduct of photosynthesis. This is where we will start to track the passage of carbon through the carbon cycle. When sunlight strikes a leaf, the energy in the sunlight is captured and used to convert 6 molecules of carbon dioxide gas from the atmosphere into the 6-carbon sugar, glucose, which can be used as a source of energy for cellular reactions. Glucose can then be stored as starch to be used later for energy, or used to make other molecules such as cellulose that form the cell wall of plant cells and contribute to the biomass of the plant. Cellulose is one of the substances aiding in the rigid nature of many plant structures, like the leaves, branches, stems, and trunk. When a log or other portion of a tree is burned, the stored glucose is converted by oxidation back to carbon dioxide gas, which is released into the atmosphere. In a similar way, the burning of fossil fuels, which contain carbon that was sequestered in prehistoric decomposing plant and animal tissues and buried for millions of years, releases carbon dioxide into the atmosphere. Animals are not autotrophs, but heterotrophs. Because heterotrophs depend on the carbon cycle to obtain energy and molecules necessary for growth, they need to consume other organisms. They do this by eating plants, or by eating other animals that eat plants. The glucose a deer acquires from eating plants contains carbon that the plant converted from carbon dioxide gas during photosynthesis. The process of converting glucose to energy, a process called oxidative respiration, uses oxygen to break down glucose molecules in cells, ultimately producing ATP, adenosine triphosphate, the energy of most cellular reactions. Carbon dioxide, generated as a waste product of the metabolic oxidation of glucose, is released into the atmosphere. Through the same process of oxidative respiration, plants also release carbon dioxide into the atmosphere. This occurs predominantly at night when they are not using carbon dioxide for photosynthesis. In addition to emitting carbon dioxide through oxidative respiration, animals release some carbon as solid waste. These solid waste products are broken down in the soil by decomposers, saprobes, and detritivores, organisms such as bacteria and fungi that degrade the waste to remove recyclable nutrients that they use for energy and growth. In the process of breaking down animal feces, decaying plants, and dead animals, decomposers also release carbon dioxide gas into the atmosphere. Carbon cycles in a similar way through aquatic ecosystems. Carbon is present in water as dissolved carbon dioxide and bicarbonate ions. Both are sources of carbon for photosynthesis and are taken up by algae and aquatic plants. Carbon dioxide is released into the water through oxidative respiration in these organisms and in other aquatic organisms such as fish, and in decomposition. Carbon dioxide is also exchanged between the water and the atmosphere. The constant absorption of carbon dioxide from the atmosphere by photosynthetic organisms and its conversion to carbohydrates by photosynthesis is key to sequestering carbon in the biotic environment. The breakdown of glucose by respiration or burning converts the carbon back to carbon dioxide, releasing it back into the atmosphere. These two processes contribute to the carbon cycle. Because there is a finite amount of carbon in the Earth's biosphere, nutrient cycling such as this is essential to life on Earth. When viewed as a biogeochemical cycle, one can see how the carbon cycle is a balance of carbon stores in the atmosphere, in the bodies of living organisms, and buried in the ground as fossil fuels. Maintaining this cycle in the closed system of the Earth's biosphere allows organisms to live on Earth and participate as key players in the carbon cycle. Very few organisms.
Alright, so bring up this idea of photoautotrophs. Alright, so your photoautotrophs utilize sunlight as an energy source. In this case, the carbon source is carbon dioxide. Alright, so they use an inorganic form of carbon. Okay. So they use this carbon source and sunlight to generate their own food. In this case, they produce mainly sugars out of this particular pathway. Now, these sugars can be used to make other things in the cell. All right, so we've already talked about during cellular respiration how various products during a stage of respiration can go on to make other larger biological molecules. It all depends on what the cell actually needs. Alright, so this particular group of organisms, alright, so it includes all three domains. So your archaea, your bacteria, and eukarya. Chemoheterotrophs. Alright, chemoheterotrophs utilize organic molecules as both a source of energy and a source of carbon. Alright, so keep in mind, the energy stored in these molecules is within the actual chemical bonds themselves that hold together the atoms in that said molecule. The carbon source is the actual organic molecule. Alright, so recall what an organic molecule is. You know, what makes up an organic molecule? Well, it has to have carbon. Alright, typically carbon and hydrogen. Okay, so your simplest organic molecule is methane, or in this case, CH4. Carbon and hydrogen. Alright, so again, all three domains of life carry out this particular process, or it can be categorized as chemoheterotrophs. Now, in terms of energy, now you could talk about the entire electromagnetic spectrum, all right, going from gamma rays all the way down to radio waves. We are going to focus our attention on only the visible light spectrum. All right, so basically the electromagnetic spectrum that we ourselves can physically see, hence the term visible light spectrum. Okay, now you'll want to familiarize yourself with the colors of the visible light spectrum. Alright, so here you go from violet to indigo to blue to green to yellow to orange to red. Alright, so violet, indigo, and blue have much shorter wavelengths. Alright, so within a period of time, going from one wavelength to the next, there's a shorter distance, okay, between these peaks. Alright, so within a certain time span, you're going to have more waves in that time span. And think of the fact that you have more waves. Think about a spring, for instance. The more coils it has, the more tightly compacted it is, closer together. When you release it, there's more energy. It's going to expand further out, right? As opposed to something like the red light, which has really long wavelengths and much lower energy. Alright, so this means within a period you have fewer waves, alright, and the distance between these waves is much greater. Alright, so now you have a slinky that is kind of partially uncoiled, and you release it, and there's much less expansion. Alright, so there's a greater amount of potential energy stored in shorter wavelength components of the visible light spectrum as opposed to longer wavelengths, colors of the visible light spectrum. Alright, so think about the visible light spectrum that we ourselves use, that our eyes use. Alright, so you're looking at the sensitivity of the human eye to particular wavelengths of light, the visible light spectrum. And you're comparing that to the visible light spectrum of that which plants utilize for photosynthesis. Alright, now notice that plants use a much larger proportion of the visible light spectrum than we do. Well, why is that the case? Well, think about it. What component or structure is responsible for detecting or responding to visible light? Well, in this case, you're talking about pigments. Right? And plants have many more different types of pigments than the human eye does. Alright, human eye, we mainly have two main types. We have our cones, got our rods. Alright, so you use cones to see color, detect color light, perceive it. And we use our rods to detect black and white and shades of gray. Okay. Now, plants have all different kinds of plant pigments. We'll talk about those in the next couple of slides. But the fact that they have different kinds of pigments allows them to use a much more wider range of the visible light spectrum. Now, notice that these pigments, collectively overall, alright, this is not a clear-cut line straight across where all those pigments use all different components of the visible light spectrum the same. Alright? In this case, different pigments are able to utilize certain wavelengths of light better than others. Alright, so in this case, this is why you see this difference in the sensitivity of plants to visible light. Alright, what components of visible light they use the best. Alright, so we'll come back to this again, but you'll notice that the blue, violet, indigo light is most utilized, and then the kind of the orange or red is. Alright, and then these areas here, they're not utilized as well in terms of green and yellow and some of the orange. We'll talk about why that is. Alright, so looking at these particular wavelengths of light, as they hit or strike the pigment molecules. All right, electrons in the pigment molecule become excited, and so they're excited to a higher energy orbital, and then they'll fall back down to the orbital they were once at. So we call this the ground state. And when they do this, you have an emission of energy. Or you have a transference of energy as this electron falls back down to ground state. Now, looking at, for instance, blue light in comparison to green light in comparison to red light. Alright, so blue light, shorter wavelength, more potential energy. It excites the electrons to a much higher energy level than does the red light. Red light, longer wavelength, less potential energy, so there's a lower amount of energy that's transferred to this electron. It's excited, but not as high of an energy state as the blue light excites the electrons. Notice here that the green light for our chlorophyll molecule, our pigment, does not excite the electrons. Now this is interesting because you don't see an absorption of energy in terms of green light. In this case, if you don't see an absorption, that must imply that you either see a transfer of light, transparency, the light passes right through, or what we really see here is the reflection of green light. Alright, so here, chlorophyll is not really good at absorbing green light, so it reflects it. So this is why we see most plants as green. Alright, so looking at this energy and what's happening when these pigment molecules are absorbing the energy from these various wavelengths of light coming from the sun. Well, one thing that can happen is that your chlorophyll molecule, the electrons are excited, they're raised to a higher energy level, they fall back down to ground state, and they release heat and fluorescence. Okay. In terms of photosynthesis, what you're going to look at here is you see this clustering of chlorophyll and beta-carotene pigment molecules. Alright, these collectively make up what's referred to as an antenna complex. We also refer to this antenna complex as a photosystem. Alright, so here, your chlorophyll molecule, this one here, is going to absorb the energy from a photon of light. Alright, so here, the electron becomes excited, is raised to a higher energy state, but in this case, instead of falling back down to ground state and releasing here energy, it's actually, the electron's actually transferred to another chlorophyll molecule, and then to another, to another, to another, and then finally is transferred to a reaction center. Alright, this particular reaction center is a special chlorophyll molecule. Alright. It's a very special chlorophyll molecule. Alright, in this case, you're moving electrons from one chlorophyll molecule to the next. All right, well, in this case, it's nothing more than a series of redox reactions. All right, so again, you're going to see oxidation reduction reactions come back. All right, so again, if you're not comfortable with these terms, you need to become comfortable with them. All right, because you're going to see it later on when we talk about the particulars or specifics of what is really going on in photosynthesis. You've got to understand oxidation and reduction. All right, so here, your little photosystem. We refer to it as an antenna complex. All right, because that is actually what it is. It sticks out above the location of where your pigment molecules are found, and we'll talk about that in the next couple of slides. And acts like basically almost like your car antenna. Right? Your car antenna receives radio waves, right? Well, this antenna receives light waves. Now, the next thing that can take place is that once your reaction center has accepted this electron, well, that means that now that is reduced, it can give up its electron to an electron acceptor. Now, this electron acceptor is actually going to be found in the membrane of thylakoid. And this will make more sense in the next couple of slides. All right, so this is kind of similarly analogous to proteins that you find in the inner membrane of the mitochondria. All right, the thylakoid also has little membrane proteins that their job is to accept electrons. All right, so looking at the anatomy of a leaf. All right, so first off, the leaf is the organ for the site of photosynthesis in plants. All right, so when you take a cross section of a leaf, all right, and you're looking at the tissue of the leaf, all right, the leaf has a top side and a bottom side. All right, now this top and bottom portion of the leaf has its own epidermis. Now, this epidermis is covered by a waxy cuticle. All right, much like our skin, the cells on the top layer of our skin are covered in this waxy cuticle. They're keratinized skin cells. Well, plants have cuticles, a waxy cuticle that's laid down on top of the surface of the leaf. All right. The function of this cuticle is that it helps to prevent water loss or desiccation. All right. So you look below the surface of the leaf. All right. You look at your mesophyll cells. All right. Your mesophyll cells are the site of photosynthesis. All right. In terms of the cells that are carrying out photosynthesis, they contain somewhere between 30 and 40 chloroplast each. All right. So the tissues of the leaf. All right. If you ever looked at a leaf, you'll notice that there are veins that are passing through this leaf. Well, the veins are made up of xylem and phloem. Xylem is responsible for transporting water. Phloem is responsible for transporting food. All right. So as opposed to our vasculature, where everything's transported in one type of vessel, the plants have it split into two different types of vasculature, one for food, one for water. Now, it doesn't mean there isn't a mixing or intermingling somewhere along the way, but right now they keep the two separate. All right, so notice how your mesophyll cells in the tissue cross-section of the leaf, some are compacted tightly with one another. All right, these are your palisade mesophyll cells. And then you have other mesophyll cells that are spongy mesophyll. Now, these are not as closely compact. All right, if you ever looked at, like, for instance, a cross section of bone, you know, like the spongy component of the bone. all right, it looks very similar. It's porous. All right, so here, these pores are useful because you can have gases that get trapped in these pores and this spongy mesophile. Now, how exactly do gases enter and exit the leaf? Well, here we have our little bitty stomata. Our stomata is made up of a pore and two cells that flank this pore that we call our guard cells. Alright, so the guard cells regulate whether or not this opening is open or closed. Alright, so depending upon whether or not, in terms of how this guard cell opens and closes, you have an influx of ions into the guard cell. Alright, guard cell swells and enlarges. You'll have an efflux of ions out. The guard cell will basically shrivel up and collapse and close. Alright, so a cell can regulate whether or not these pores are opened or closed. Alright, so gases like CO2 enter through the stoma, gases like oxygen exit, as well as water. Alright, so if you think back to the chapter over water, alright, it talked about transpiration. Well, keep in mind that water molecules are both cohesive and adhesive in nature, and that when you're talking about the surface of a leaf, these pores are open, water molecules near that pore begin to evaporate. And then water molecules attached to that water molecule is evaporated, gets pulled up. Alright, so this is how water is pulled up to the leaves. It's through this process of transpiration. Okay. Alright, so looking at the mesophyll cells themselves. Alright, so notice here you're zeroing in on just a single organelle. Alright, the chloroplast. Alright, you'll want to review the structure of the chloroplast again. Alright, because you'll want to be familiar with it. So here, your chloroplast has an outer inner membrane. It has this internal cavity, sac, that contains a fluid-like substance that we refer to as the stroma. And then you have these discs. And these discs are referred to as thylakoids. Alright, now, when you talk about these little discs, you have a stack of them. You're talking about a singular stack, you're talking about granum. If you're talking about it being many stacks, talking about grana. And here, looking at your thylakoid, it's not some just empty sac. Alright, it has its own membrane, and in the membrane, you have your pigment molecules, like chlorophyll. Alright, in which your little chlorophyll pigment molecules embedded in the membrane of the thylakoid. And then inside of this disc, you have this cavity, this lumen, that we refer to as the thylakoid space. Alright, so looking at our photosynthetic pigments. Well, the pigment molecules themselves, for instance, looking at chlorophyll A and B. Well, you have this porphyrin ring structure that looks a whole lot like hemoglobin in terms of structure, porphyrin ring. Alright, and then you have this long hydrocarbon tail. Well, what other molecules have hydrocarbon tails? Well, your fatty acids do. And hydrocarbon tails, like fatty acids, are they polar or are they non-polar? Well, in this case, they're not polar. They're non-polar. Again, recall the difference between a polar covalent bond and a non-polar covalent bond, and how that affects the structure itself, whether or not it's polar or non-polar, how it affects where it's located in a cell. Alright, so this non-polar hydrocarbon tail, it's going to be stuck in the membrane, the fatty acid portion of the thylakoid membrane. And then your porphyrin ring structure, all right, the antenna or antennae portion is responsible for absorbing light energy. So it sticks out above the surface of this little thylakoid disc.
The thylakoid membrane in chloroplasts contains light-absorbing molecules called pigments. Different pigments absorb light of different wavelengths. One pigment, chlorophyll A, absorbs mainly blue-violet and red light. This molecule participates directly in the light reactions. Another molecule, chlorophyll B, absorbs mainly blue and orange light. We do not see these absorbed colors when we look at a leaf. Instead, we see the green wavelengths that are reflected back from the leaf or transmitted through it.
Alright, so here you're being shown a chloroplast, and you're looking at the little thylakoids, and the thylakoids are absorbing the light energy. Specifically, the pigment molecules like chlorophyll are absorbing the light energy. And here you have the indigo, the violet, the blue, the yellow, the orange, and red light that's being absorbed. Here you have green light that's either passing through or it's being reflected. So the reason we see and perceive leaves, most leaves, as being green is because they reflect green light. They don't absorb it. Again, this is tied into the fact that the chlorophyll molecule itself does not absorb green light readily. Alright, so how do we know exactly what type of plant pigment absorbs what type of light and reflects other kinds of light? Well, you can use a device called a spectrophotometer. Alright, so all that means is that you can look at a variety of spectrums of the visible light spectrum. And you can look at measuring what light's absorbed or transmitted through a solution. Alright, so you have a light source, alright, like a light bulb. You have a prism that the light passes through, where you get all the different colors of the visible light spectrum, and you can filter what light you want to hit your sample. Alright, you can ultimately determine what wavelengths of light you want to pass through your sample. Okay. Now these wavelengths of light pass through your sample, for instance your solution of chlorophyll, and then you have a device within the spectrophotometer that's responsible for detecting what light, how much light, actually passes through your sample. So here you're looking at 100% of the light that passes through the sample is being detected. Alright, so you have 100% transmittance. So there's very little light that's actually being absorbed by the chlorophyll in the solution. Alright, here looking at blue light, the light passes through the sample, and you'll notice that there's very low transmission of blue light. Alright, a majority of the blue light that passes through the sample is absorbed by the chlorophyll molecules. Very little passes through and is detected. Alright, so in this case, high transmittance, low absorption. High absorption, low transmittance. Alright, so we can put this to use. We can develop a graphical representation of the different wavelengths of light that each particular pigment molecule utilizes, and how well it utilizes that particular wavelength of light. And we could tie this in with photosynthesis and the rate of photosynthesis, and which wavelengths of light are most utilized for photosynthesis and which are not. Alright, so here you are looking at, again, chlorophyll molecules A and B, again, you have slight changes in chemical groups, functional groups, all right, which affect what type of pigment molecule you're dealing with. And then you have beta carotene. All right, so again, a long hydrocarbon tail. All right. So here, looking at the visible light spectrum. All right, so you're looking at an absorption spectrum here for chlorophyll A in green. So it absorbs indigo, violet, and blue light pretty well. It doesn't absorb blue, green, or yellow, or orange very well. Red, orange, and red it absorbs pretty readily. Looking at chlorophyll B, there's a shift. All right, it absorbs the blue, green, and some of the green light fairly readily, and then some of the orange light and orange red. All right, neither of these two absorb green light and yellow green light readily. All right, so again, this is why when we look at a leaf, we see green, yellowish colored leaves. All right, because those particular wavelengths of light are not absorbed by these particular pigment molecules. Now, what happens during the fall season? Well, your chlorophyll molecules begin to be broken down, right? And what you're left with are other pigment molecules that participate in photosynthesis. All right, but they absorb light, visible light, at other parts of the spectrum. All right, so carotenoids like beta carotene absorbs blue, green, and green light pretty readily. Some of the purple, some of the blue, indigo light, but it doesn't absorb yellow, orange, red, or red light. All right, so this is why leaves during the fall, you begin to see yellow, orange, red, yellow, orange colored leaves. All right, because the pigment molecule that makes up a majority of the pigments found in the chloroplast is now carotenoids as opposed to chlorophylls. All right, so this is showing you the action spectrum of photosynthesis. All right, and tying this in with the absorption spectrum. All right, so notice that the absorption spectrum of light, so chlorophyll A and B is here and here. All right, so mainly you're looking at indigo, blue, and light blue light, and then orange, red, and red light as being the major players involved in what they contribute to photosynthesis. All right, those particular wavelengths provide the most energy. All right, and then your green light and your light blue, light green light, light blue green light, and some of your yellows don't contribute as much. All right, they're not as absorbed readily. All right, so they don't give as much energy, overall energy, for photosynthesis. Now, some of your carotenoids do absorb some of the green light, all right, but you do have this dip here, which is mainly your green light and yellow light. So it makes sense that those particular wavelengths of light that are absorbed the highest and most efficiently contribute the most to the rate of photosynthesis. Alright, so how exactly do we know which wavelengths of light found in white light, alright, all the different colors of the rainbow, are responsible for driving photosynthesis? Well, here you have some algae, and you expose the algae cells to all wavelengths of visible light spectrum. Alright, you can use a prism. And then you have bacteria that seek out the presence of oxygen, all right, because your algal cells that are undergoing photosynthesis are going to release oxygen. All right, so at a particular wavelength of light that's most utilized, they're going to release more oxygen. All right, so these bacteria are going to congregate and associate around that particular algal cell is exposed to that particular wavelength of light that produces the most oxygen, that increases the rate of photosynthesis the highest. Alright, so we know that here are indigo, purple, blue light, and orange, and red light contributing most to photosynthesis. So here, the bacteria are congregating around those particular algal cells. So those wavelengths of light provide the most energy to drive photosynthetic reactions. Alright, so photosynthesis, again, is a redox reaction. Alright, so here, carbon dioxide is going to be reduced to a sugar. Alright, so in this case we have glucose. Water is going to be oxidized to oxygen gas. Alright, so here the plant is taking in inorganic carbon dioxide, inorganic form of carbon, taking in water, using light energy to drive the synthesis of sugars, and as a result you have the production of water and oxygen gas. Alright, so the CO2 is going to pick up electrons and hydrogen atoms from water, and those are going to be contributed into making the sugar. And then you're going to have the oxygen that was in the water molecule itself. The oxygen atom is going to be released, and so it's oxidized to oxygen gas. Alright, so looking at how this particular process of photosynthesis, the reaction, where each individual atom as a reactant or component of the reactant molecules goes into the final products. Alright, so here your 6 carbon dioxide molecules. Those 6 carbon atoms become the 6 carbon atoms of your sugar. Okay, so it's a nice balanced reaction. Alright, the oxygen atoms become a component of the sugar as well as any water you make. The water, as a reactant, contributes hydrogen atoms to both the sugar and the water that you make, and the oxygen that's in the water is released as oxygen gas. Alright, so this is the oxygen gas that we all breathe. This is the oxygen gas that plants produce. And this is how they produce oxygen gas, is they split a water molecule. Alright, so this is kind of setting you up for photosynthesis and looking at specific reactions of photosynthesis. Alright, so here we're looking at our chloroplast. Alright, you're looking at your thylakoid membrane, and you're looking at your stroma. Alright, so the light reaction occurs in the thylakoid membrane, and your, what we call archaically, the dark reaction. It's more appropriately termed the light independent reaction, or better known as the Calvin cycle, that occurs in the stroma. Alright, your light dependent reaction, your light reaction. Alright, your pigment molecules absorb light energy. That light energy excites electrons. Those electrons are used to reduce NADP plus to NADPH. They're also used to generate ATP. Now, these electrons are lost in the process. Alright, they get transferred to these electron carrier molecules here. So they have to be replaced. All right, so this is where water comes in. Water split, it donates electrons back to your pigment molecules, and you produce oxygen gas, which leaves the cell through the stomata. And then we breathe it in. All right, now you make all this ATP and you make all this NADPH out of the light reaction. All right, you have your CO2 gas that comes in. The CO2 gas is going to be reduced to a sugar. All right, so in this case, glucose, and more specifically, sucrose, mainly, in plants. All right, so you use the energy, the stored potential energy in these molecules to reduce CO2 to sugars.